Hi everyone, and welcome to Let's Talk Risk with Dr. Naveen Agarwal, where each week we talk about a topic related to risk management of medical devices. I'm your host, Naveen Agarwal, principal and founder at Achieve, where my personal mission is to help you achieve success in risk management. In this episode, I'm joined by Sri Kaushik to talk about risk management considerations for in vitro diagnostic devices. She has years of experience, both on the medical device side and the IVD side, to be able to understand the subtle differences and challenges involved in risk management of IVDs. We talked about this topic as part of a LinkedIn live audio event in front of a live audience. You're about to hear a recording of our discussion. So sit back, relax, and enjoy the show. I see a lot of familiar faces in the audience today. Ricardo, Leo, Joshua, Andy, Vijal, Christy, thanks for coming back. And I see a lot of new faces as well. So welcome to all of you. It's just great to be back with you again. And during our conversation also, please continue to share how you are feeling and how you are thinking about the discussion using the react button. So with that, Sri, I want to welcome. Thanks for joining again.
Thank you very much for having me, Naveen.
So Sri, let's just start by sharing a little bit of your personal story with the audience and tell us, how you reached to the place that you are right now in your career. What have been some of your pivotal moments in the career that you have? You have a very, very impressive background and experience. So tell us a little bit more about what your journey has been to this point.
Thank you, Naveen. So I want to begin with a quote that Mark Twain made, which is, to succeed in life, you need two things, ignorance and confidence. And I have an abundance of both. And so I am really successful in what I do. That's supposed to be a joke, people. Please take it that way. And 30 plus years, I worked on the bench and doing research, thought that I was going to be a faculty member. And so one fine morning realized that nobody wanted me to be a faculty member, and rightfully so. And pivoted out, and of all odd things, I was a biochemist, molecular biologist. I was thinking about drug regulatory and stuff like that. And then I read oddly a business news article, of all things, that talked about personalized medicine and how diagnostics are helping people overcome simple things like height problems, or just kids growing taller and stuff like that. And it sort of put me into a path of wanting to understand what personalized medicine was. This was way back in 2008. Lo and behold, I pivoted out directly out of my bench work into submitting 510Ks for medical devices to FDA. And I got a break because I had a phenomenal mentor who wanted me to come in and just help him and paid me for what I was doing. And we did about 12 510Ks together very quickly, all of them in the medical device realm. But I've always wanted to be in IVDs. And Joshua, who's on this call, was the first guy who told me how I can learn about IVDs. And this was back in 2013, and I've been learning ever since. And I got an opportunity to switch to the IVD world and when I joined as a VP of regulatory for a company that I was helping in the early stages in Baltimore, just pro bono, because I really wanted to get in the field. And it turned out that that's where I had I don't want to say a rude awakening, but it was more of an awakening of sorts, because I'd known medical devices, and I'd sort of written risk management documents and submitted as part of my 510Ks, and I thought I understood a medical device is a medical device, and an IVD is a medical device, so there's nothing much different to it. And then I walk into that realm and suddenly realize, oh my God, this is so much more different, right? It's different because medical devices actually touch the patient, and they actually are you can sort of work through the risk process and mitigate the risks because you know that the end user is actually going to use your device.
I see.
In an IVD realm, your end user is somebody who just runs the assay and gives that information to whoever, right? It could be a clinician, it could be a clinical lab, it could be the patient.
Right.
And what they do with that information has nothing to do, at that point it becomes clinical practice, right? And a doctor may run a test but do nothing about it. And so how do you mitigate that risk? And how do you deal with risks that come out of that? And that was something that I had to learn.
So what you're talking about really is the diagnostic device is providing us some information that should be used to make a clinical decision. So there's a step,
not necessarily.
Right?
Not necessarily. There's very good reasons why you don't want to make clinical decisions, right? So let me give you a personal example from COVID. Last year, my mom lives with me. She's 75, and my entire family came down with COVID. And just weeks before that, a friend of mine got COVID, and she went to her doctor, and the doctor gave her the medicine to stop COVID. Right? I mean, I don't remember the name of the medication, but she was given that. And she said she got better in 2 days. So as soon as my mom got COVID with all her maladies, I called my doctor and said, hey, can you prescribe the medicine? My doctor said, no. And I said, okay, I trust you, man, because you've been my doctor for 15 years. I will just let it be. But my mom was suffering for 7-8 days consistently. Then after the 5th day, I called him and said, what is going on, dude? Everybody else is giving this medicine. Why aren't you? He said, sometimes the relapse from the drug is worse, and she would have suffered a lot more if I had prescribed the drug to her. Than not giving her the drug. So true enough, this friend of mine, I talked to her a month later, and she said, yeah, I got COVID back so bad the second time after the drug regime was done, that I was down and out. The first time I wasn't that sick. The second time I was down and out. So the rebound of the drug is a humongous risk, and doctors should have the ability to say, no, we're not giving you the medicine.
Oh, even though the test is showing positive.
Yes.
Okay, so what you're talking about really is a very interesting case. And first of all, I'm so glad, that everyone is good. They're healthy. COVID was a bad experience for all of us. But it basically helps me put this in context when we talk about risk related to IVD, because we have to build sequence of events, what might come out of it. And in this case, you are saying the clinician made a very good judgment based on benefit risk using their experience with other patients. And the relapse was a much bigger risk than actually. So they must have, by the way, thought about an individual patient. And in that context, they made that decision, right?
Yes, and he basically didn't give it to me, or he didn't give it to my wife or anybody, because he said, I don't want you guys to suffer. And that's a great doctor, first off. And the second part was essentially that, do all IVDs, are all IVDs the same way? That, there should not be an intervention. I would never say that. Personalized medicine is the reason I got into IVD field. And in that particular case, if you can mitigate some of the risks associated with devices, especially in the oncology space, so with drugs in the oncology space, I think a diagnostic is very useful. Similarly, for certain infectious diseases that can be cured. I mean, you don't want somebody to suffer through anthrax because there's antibiotics available to treat those.
Nice.
If you are diagnosed with anthrax. So it's an indirect way in which risk affects IVDs. And usually it is false negatives and false positives that are the most critical items that we as a regulatory person developing IVDs we have to worry about.
Got it. So Shri, I'm going to just pause for a second, because I know I was not aware of the quality criteria and performance criteria for IVDs. Would you mind very briefly describing what are the critical criteria or parameters? You mentioned false positive, false negative. I wasn't aware myself, so I'm hoping that some people can actually benefit from this, just basic information first. Would you mind doing that?
Yeah. So, the intent of most IVDs is, at least this is my opinion, there's much better IVD folks on this call that can probably correct me. The intent of the IVD is to diagnose a disease state, right? And, ideally you'd want to, when you have the disease, you want 100% of the times for the IVD to be positive. And when you don't have the disease, you want to be 100% of the time sure that the IVDs are, that you don't have the disease.
Got it.
So that's specificity and sensitivity, which is what you get to hear a lot about. However, we being complex people, those things are not that distinct, because it's a biological process, and biological process has inherently, what works for me won't work for you pretty much, because of my genetics and my physiology and my, background of how I have grown up and versus how you've grown up. And so our phenotype and genotype will dictate whether or not we're going to respond to something correctly. Though, you would definitely agree that you don't want to be prescribed a drug that could potentially have, all drugs are going to have some side effect, because it's a foreign product or any therapeutic is going to have a side effect. You don't want to take that side effect if you can avoid it, right? And so from a quality perspective, what you definitely need to do is that you have to make sure that your IVD meets the user needs, and you have to be able to weave your risk assessment, user needs, into your risk assessments from a design input perspective. So your design input should inform you as to what level of risk mitigation you need to put for each and every step. Got it. And as an example, if you want to say, I have a COVID test, but I only can detect 80% of the times that this person probably has COVID. And in today's date, and this is not a political statement, but we know that the vaccines that were approved, at least for people under 25, in boys, in men, is giving myocarditis. Do you want to take that risk just because, and those kinds of decisions have to be made in the context of the science and the user need. If I was developing an IVD, I would say, and this is not the right way to do it, but this is just a thought, which is I wouldn't let any boy under 25 actually use an IVD test because of the fear of somebody for COVID, because of the fear that somebody might say, go take a vaccine.
Got it. So, of course, I think what we are also saying is that these decisions are made in a clinical context with a specific patient. And what I'm hearing you say is that the IVD should be accurate enough to drive the right decision in a clinical context.
Yes.
That's the purpose of the IVD, right? And that's why false positive, false negative matters, because if we are getting a false result, we have a risk of either delaying the treatment or using an incorrect treatment that may be harmful to the patient, right? I think that's the main idea. So many things happened during COVID. I know, and I have read up a lot on this. I have been very, very nicely informed and educated. There's a lot of conversation because we are still learning about it. So here's what I would say, Shri. I think this was a very good kind of a segue here for now inviting our audience to participate. And maybe, first of all, I want to recognize a few things. You mentioned Joshua is in the audience. Josh, hello. In fact, Josh, I think you are the reason that I'm talking to Shri today, because you helped this kind of networking and relationship building. So I want all of us in the audience today to understand the value of networking. And if you guys didn't know, you can connect with anybody one-to-one in this audience today, even if they are not your first degree contact. So you can look up their profile and, just don't spam anybody. Let me just qualify that. But what we are also trying to do is encourage participation and networking. Okay, so this is how it's going to work. Those of you who are new, you may not have known this, but if you want to participate in the discussion, which I heavily encourage you to do, just please raise your hand and you will find that at the top of your profile picture. Just request an invitation to speak and join Shri and I on this, what I call virtual stage. And those of you who have been here before, you know how the process works. So I hope to see some hands going up in the air right now, because I would love to have you guys participate and share your thoughts. As Shri mentioned, we are all learning from each other. And I believe you have a lot to contribute. And you might have some interesting questions to ask. So I invite you at this point of time to somehow indicate to me that you are interested. So let's wait to hear from our folks in the audience today, Shri, but let's continue our discussion. So we were talking about why false positive, false negatives results are important, and we must control that. So do you have some sort of benchmarks in mind, some IVD case examples where you would say certain level of accuracy must be demonstrated reliably before we can go forward? How do you decide that?
So, for instance, if you're talking about Red Cross takes a lot of blood for blood transfusions, right?
Mm-hmm.
Most diagnostics that are used for blood screening for transfer, they are considered to be highly risky, because when you take blood, sometimes these companies basically don't just transfer whole blood. Sometimes it's plasma. Sometimes you take plasma out, and you can do it, and you can just give buffy coat or something can be taken out and injected into a patient. So one bag of blood could infect a ton of people.
Yeah.
So in that particular case, and rightfully so, CBER, which controls the blood group, they require blood diagnostics to be really, really high sensitive, highly specific.
What does that mean? So, what does really high mean?
So it's 99% plus or minus, with a lower CI of 98.
Oh.
So it essentially means that you need to be 99% sure that if you say if somebody's blood doesn't have HIV, that it doesn't have HIV, right? That is true for a lot of those, even the hepatitis and all these other devices, IVDs. However, if you're just, if the doctor is doing a test in the hospital, you walk in looking yellow, and they want to run a test for hepatitis B or C. That could potentially have a slightly lower sensitivity requirements, which would be 95% or with a lower CI of 90, which essentially means that 95% of if you took 100 people, 95% of the times my IVD will say this is correct. So depending on the use scenario, the IVD folks actually sort of have to really weigh in what the consequence of it is, and a manufacturer has to mitigate risks of false positives and false negatives.
So
tightly.
Yeah. So what you are saying is that it's a risk-based decision. What the design requirement for the IVD would be, right?
Yes.
Wow.
Yes, it is.
So then
it's always a risk-based decision.
So yes. then I would expect a very tight kind of hand-holding or integration between risk analysis early on and design inputs early on. How does that happen? Can you share some insights on that?
You would want that, Naveen, but my experience has told me that usually, at least risk in itself is not the top priority of development. So the biggest issue in 24971 actually says that you need to have user needs as design inputs to be able to do a good risk assessment and risk management, right? Now, if that's the case, then everybody should be working on user needs. Most startup companies, small companies, don't have very well-defined user needs. And because of that, your risk is always lacking. But ideally, the way it needs to be done is the same way quality is dealt with in the device world, which is the management has to buy in, and the buy-in can be facilitated, in my opinion, by two ways. Either the management is conscientious that they want to do it, or they need to be told that
some information.
The worst thing that can happen to a company is not FDA giving a rejection on their application, because those things can be mitigated with the correct testing, but loss of revenue.
Yeah, but
shutting
Sri,
down.
So this is so amazing and such a coincidence. And guys, this was not planned. I'm so glad to welcome Christy here on our virtual stage. Christy, thanks for joining. You work with small startups all the time. And she mentioned this challenge. I would love to hear your view. How do you help them understand this basic requirement?
The basic requirement to link user needs to
and risk management. Yeah.
I honestly, I teach risk management like storytelling. And the story really comes back to what's driving the risk? What's the foundation of the risk existing in the first place, which sometimes comes from user needs. And then after like the climax of the story, right? Like after you've identified the potential for harm, what do you do about it? And sometimes that turns into a new user need that you didn't know you needed. So that's how I break it down for little teams. I wanted to actually come on to ask Shri about your experience with 24971 Annex H. I remember it because H is happy for me. Annex H it's seriously, I'm like, risk management is such a fun area. It's kind of like a sickness. Like my business partner tells me I have a sickness because I love it so much. But I help a lot of really, really tiny diagnostic teams set up their risk management files. And we start with ISO TR 24971 Annex H, which my friend Ed Bills would tell you is a gift from the ISO committee, which it really is, because it's like 22 pages of goodness that's all about IVDs, right? But my question for you is, what other resources do you have, or how do you suggest for really small teams in the diagnostic space to get started? Because a lot of times it's like, two PhDs out of MIT with a really cool idea. So I try and break it down and turn it into, easy to understand terms. But Annex H is both gorgeous and there's a ton of information in there. So I'm wondering if you have any best practices for how you break it down for the little guys.
Nice. Thank you, Christy. Go ahead, Shri.
Thank you very much. And I can tell you the first thing, and Ed Bills is the one who taught me my first foray into, risk analysis. And I took the AMI course, and I took it because I wanted to basically learn how to do the IVD regulations, right? And that was just, I think, 3 months or 4 months before that TIR 24971 was released. And one of the things that happened was whenever I've gone and talked to any of these meetings, everybody talks about medical devices, and then they say, IVDs fall into the same category. Well, it doesn't. And it really doesn't. I mean, the IVDs have its own little world, and so Ed pointed us to the H, and I the way I agree with you. It is a work of art, and it's sort of all the questions and all the issues, and for instance, like we do the P1, P2. Assessment in when you're sort of categorizing the risk, and you have to take into what the manufacturer can do as P1, and P2 is what the users would do with your device, and what's the risk to the users for the users. And the first time I ever figured out that P2 does not necessarily apply to IVDs was when I read H. Right? And so it was not only eye-opening. And if somebody wants to get into IVD regulatory, I would strongly suggest go pay the money. It's not that expensive. It's like $270. Get TIR 24971, open up H, and then sort of go through line by line what they say. And I really, really do agree with you because that is definitely a gift to IVD folks. Because
so are they?
Till then, I never got any IVD. Most people everywhere talk about medical devices and then IVD.
So Shri, very quickly then, because I know Roger has probably a question or comment. Very quickly, are there any other resources that you would recommend to people just starting out?
Yeah, so one of the places I actually do a lot of searching on is the medical device reports database from FDA, MDR reports. Usually what happens is when you have an IVD that, could if you have something similar to your device in the MDR, the FDA has this TPLC page where you can just go click and then it tells you all the risks all the failures that have happened. Mm-hmm. And usually you could go in there and if you can find something useful within your own development process, I frequently visit the MDR database for potential risk inputs and if my device can have the same issue. I do a lot of Google searching and I do a lot of reading on similar devices. So if I'm using a blood diagnostic, I would go download something from Glucose Diagnostics or some kind of other blood diagnostic where you take a finger stick and see what kind of risks are happening with those being false for false positives and false negatives.
Great.
There's various places you pull this information from. It's not just one.
Sure thing.
But I think Annex H is God's gift, ISO's gift to IVD regulatory folks.
Thank you. With that, Roger, I know you have been patiently waiting. I appreciate that. Please go ahead. What you have in mind?
I think this is a great discussion. I've personally not worked in IVDs myself and never really thought about the things that Shri brought up. But what I wanted to say, if other people are like me and kind of interested in this, there's a book I read years ago, and I don't remember the title. I don't remember the author. The title was called Intuitive Biostatistics. And I got that book and was reading it for the statistical end of it, but I'm thinking back on it. Many of the examples are set up from a clinical viewpoint, kind of this what can go wrong with clinical decisions and the statistics behind them. But I thought about it. I thought back to that book as Shri was describing some of these examples. So I'm just putting that out there. If other people maybe are interested in this, you might look into that book. I myself will pull it down and be thinking of it now in terms of an IVD rather than the pure statistics.
Perfect. Thank you.
So that was all I wanted to say.
Thank you, Roger.
Can you tell me to repeat the name of the book? I want to take a look at it.
Okay. It's Intuitive Biostatistics.
And guys, what I would recommend, first of all, Roger, thank you for sharing. And this is the beauty of our platform today, this conversation. We learn from each other. If somebody can find that link, please drop it in the comment section for this event. And that way, everybody will be able to share that. Thank you, sir. Thank you, Roger. Ed, I'm so happy to welcome you to our virtual stage. Thank you so much for joining. Please share what you have in mind.
I'm just so happy to be here with everyone and with this great audience, but also these great speakers. And I was happy to hear the reference to ISO TR 24971 and the indication that anybody in IVD should get it. Well, I will go further. Anyone in risk management for medical devices should have a copy of that manual. It's 85 pages of the latest updated information from some real experts on medical device risk management. And just about any question you might have, there's at least something that touches on it. It may not completely answer your question, but it will give you a start on research and trying to find out what's going on. And it was entirely updated when it was released in 2020. It's all new stuff. It also introduces cybersecurity and data security as elements that need to be considered. But now today, we've gone beyond that, and we now have AI and machine learning going on, and that needs to be incorporated into your risk management process. and document it in your risk management file. So many times these things grow separately, and we tend to develop them as separate issues. But in the end, all of the risks for our devices need to be part of the overall residual risk analysis. And the way you do that is by getting them into your risk management file. Mm-hmm. So don't forget, as these new things come up, to bring them in. I'm sure our next edition of 24971 will have a discussion of that. In the meantime, there's a consensus report, CR 34971. Notice how those numbers seem to follow there, which is on AI and machine learning. So there's more risk that we need to pull into the picture here, and we need to learn every day. As Roger was indicating there, there's lots to learn out there, and we need to do that.
Thank you.
I'll shut up now.
No, thank you so much. Guys, like always, we need more time. We run out of time, I know, and it's 11:30. But it's all great, right? This engagement is awesome. I'm deeply energized by it, and I'm so happy to see all of you again. I want to very quickly mention a few things to you, and then I'll open just a little bit, a couple of minutes for Shri for closing thoughts. Key housekeeping point is that in case you miss these events, I'm going to be producing the recording of our past discussions with an article describing key highlights. And you can find that on my newsletter. If you go to my profile in the featured section, the first link will bring you to my newsletter. We are going to be publishing these recordings. So in that case, you can catch up on what we are talking about in these conversations. With that, I'm going to really give a couple of minutes to Shri for some closing thoughts. Shri, please tell us what you're thinking right now.
Just thinking over the process of from where I got from to where I am, and I feel like while we're talking about risk, I think having good mentors is really worth the effort and cultivate relationships. Like, Naveen calling me on this phone call, asking me to join in. I would have never volunteered. I told him that, and but it was Naveen, so I had to come and talk about my experiences. And similarly, Josh Levin, who's left, he put me. I wanted to be in IVDs, and he put me on the path to IVDs. And he's been a friend and mentor for a long time. So find the best. When you find good people, cultivate relationships with them, because that's your risk mitigation towards failing in your careers and work moving forward. And every once in a while, when you have an opportunity, feel free to come and talk about your experience, because I love learning from people. So
that's such an amazing thought to close our conversation with today, Shri. I really appreciate that. That's probably the best advice you can give to everybody is, work hard, look up these things, learn yourself, but also cultivate mentors. And with that, I'm going to thank all of you once again for joining. Thank you, Ed. Thank you, Shri, Christy, and Roger for participating. Guys, if I had some giveaways to give you to encourage participation, I will do that. But this is a virtual forum. So you'll just have my best wishes and gratitude if you decide to participate, because that's what it's all about. Thank you guys again. We will connect once more next week. Please mark it on your calendars, 11 a.m. Eastern every Friday. And if you miss it, you will find the recordings and key highlights on my newsletter. With that, everybody have a good weekend ahead, and we will be in touch soon. Bye-bye.
Thank you.
Thanks, Naveen.
Thank you. Thank you, guys. Thank you, Shri and Ed. Bye-bye.